Next is your diuretics. Diuretics, they act at the nephrons. The first member is your osmotic diuretic, that is mannitol. That is your mannitol. Mannitol attract water. This is the mannitol. It is filtered and it attracts the water. Diuresis. So diuretics, the first category is osmotic diuretics. Mannitol. Mannitol function is to attract water. If it is filtered in the lumen, it will attract water and cause the diuresis. This is the simple mechanism of the mannitol. So this drug will not act increase the volume of urine. That is diuresis. Second group of drug acting on PCT. They are carbonic acid inhibitors. They are carbonic acid inhibitors. In case of drugs, this is the lumen of your sodium, bicarb. These molecules are filtered, and this is the PCT cell. Sodium enter inside and proton comes outside. This proton combine with the bicarb form H2CO3, that is your H2O and CO2, with the help of carbonic anhydride. Again combine with water, form H2CO3, that will form H plus and bicarb. Bicarb will be absorbed in the blood, and here sodium also absorbed. This mechanism
Hello, sir. We can't hear you. Hello. Hello, sir.
So the first category is carbonic anhydrase inhibitors. This is our nephron where these are the PCT cells. At this location we have carbonic anhydrase enzyme. Normally in the glomerulus, sodium, bicarb, these things filter and they reabsorb. But if we block this, then this sodium and bicarb, it will not be filtered or not be reabsorbed, it will lose in the urine. So in the urine, your alkali bicarbonate is lost. And second thing is sodium. Because of loss of alkali, what occur in the blood? Acidosis, that is metabolic acidosis. Metabolic acidosis. So to compensate this loss of alkali, because of this metabolic acidosis, body will compensate. It will stop bicarb filtration. It will stop bicarb filtration. So if bicarbonate is not coming, then at the level of PCT, if you are giving your drug carbonic anhydrase inhibitor, if bicarbonate is not filtered, then at this level your mechanism will stop on its own. So we call this self-limiting
Hello, sir. We have lost your audio and video, sir.
from the same channel, they are reabsorbed. If loop diuretics like your drug furosemide this channel, then sodium will be lost, potassium as well as chloride. These ions will be lost in the urine. So loop diuretics they causes the loss of potassium, sodium, chloride and calcium also. How the calcium is lost? Suppose this is the calcium molecules. These calcium or magnesium they enter in between the cells and our drug loop diuretics they block this channel so sodium, chloride and potassium all are here because they are absorbed reabsorbed because of some negative charges they comes inside but in this case these are repelled by the positive charges so they will not be absorbed in the urine you will find loss of calcium loss of magnesium so this is the most important point with the loop diuretic loop loses calcium magnesium. So PCT carbonic anhydrase inhibitors then thick ascending loop diuretics this part we have thiazide drug we have thiazide drugs there is one channel transporter that is known as sodium chloride symporter. If you block this one, then sodium will be lost in the urine. Chloride will not be absorbed. It will also come out. So thiazide, they are responsible for the loss of sodium and your molecule chloride. But in this case, there is no loss of calcium retained inside the body. Then we have potassium sparing diuretics. Sparing means they will try to save potassium inside the body. These drugs they act at the collecting duct. So if this is the nephron, PCT act here carbonic anhydrase inhibitor acetazolamide and at this location furosemide, this location thiazide and at this part we have drug known as aldosterone receptor antagonist. That is your spironolactone. These are potassium-sparing diuretics. Potassium-sparing diuretics. Spironolactone. The mechanism of this spironolactone is this is the cell of collecting duct.
Sir, we can't hear you. In potassium-sparing drug, there is one more category that is known as epithelial sodium channel blocker. These are sodium channel, these are potassium and proton. Epithelial sodium channel blocker drug. If you block the sodium channel, then it will come into the urine. But these two channels, they act under the influence of sodium channel. If you block this, they will also not work. So potassium will remain inside and proton will remain inside, and it will enter into the blood. So this is known as potassium sparing action. But the mechanism is through the blockage of epithelial sodium channel.
because these are under the influence of this channel. If you block this, they are also blocked. So potassium will be saved and enter into blood. And the second mechanism was the through your aldosterone receptor. So aldosterone enter inside cell, activate the receptor enter into the nucleus, gene transcription, protein synthesis, then sodium, potassium, proton, all channels are implanted, and then it will reabsorb. But if we block this pathway, again these two channels or the all the three are not implanted. Sodium will lost into urine and potassium they will not come out. So they will enter into the blood. So in these two ways we have saved our potassium. So they are called potassium sparing diuretic. So in case of diuretic, if we summarize all the things, first was osmotic diuretic, your mannitol. The mechanism of mannitol is very simple. Attract the water and remove the water through the urine. Second was carbonic acid inhibitors drug, acetazolamide. Third was loop diuretic, your furosemide. Fourth, thiazide. And last, potassium sparing diuretic. So one, 2, 3, 4, 5. There are total 5 diuretics: osmotic, yeah, stazolamide, furosemide, thiazide, and potassium sparing diuretic.
So,
हाँ,
okay, please
hello.
repeat potassium mechanism.
Potassium sparing one.
Yeah, the
Okay,
last one.
okay, okay.
You actually, sir.
This is the collecting duct. This is the collecting duct. At this part, potassium sparing diuretic act. There are two ways. With the help of one diagram, I will explain. Suppose this is the cell of your collecting duct. Similar cell, both are collecting ducts. There is one receptor that is known as aldosterone receptor. This is a steroid receptor. It will enter into nucleus, DNA, then messenger RNA, then formation of some protein which are called channels. If this implant three channel sodium, potassium and proton. Will work. This is normal case. If we block the receptor with the drug spironolactone, then there will not be any transcription, translation, or no channel implantation. So if there is no potassium channel, so potassium will not be able to come out from this side. So this potassium, there is one pump sodium potassium ATPase. With the help of this,
the potassium will enter into blood. So in this way, we have spared or saved our potassium. We block the receptor, no transcription, no translation, and there is no potassium channel at the lumen area. So it will not come out. So all the potassium through these channel will enter into blood and it will remain in the blood. One more category of drug we have. These drugs are known as ENA blocker, epithelial sodium channel blocker. If you block the sodium channel, then these two are under the influence of sodium. They are also blocked automatically. Then potassium will not come out into lumen or urine. Then into the blood. This is how in both the mechanism from the intracellular potassium is moved into the so potassium is not losing in the urine, so we call this potassium sparing diuretic. One is working with the help of receptor, which receptor your aldosterone receptor, and the drug is spironolactone. And the another one is just acting through the epithelial sodium channel. Epithelial sodium channel. And this drug, this is known as triamterene, triamterene, and this is spironolactone. So these are potassium sparing or potassium retaining diuretics working through two kind of mechanism. One is through aldosterone receptor and one is direct inhibitor of your epithelial sodium channel. One more point with mannitol. Mannitol is entered here. This is the compartment of brain where which is filled with the water because there is cerebral edema, lot of water. But the property of mannitol is this that it will not come out from the capillary. Still present within the capillary, it will attract water. Then your autonomic system and